right, hello and welcome everyone to today's presentation and webinar on the handling of missing data in clinical trials for non-statisticians. My name is Jim Townsend. I'm the Vice President of Business Development at STC. Thank you for taking the time out of your busy schedules, both for those of you joining us here in Chicago as well as all of you joining via webinar. I'll just be kicking things off today briefly, going over some housekeeping items, then introducing our presenters so we can dive right into the presentation. For the housekeeping items, we're scheduled for 1 hour presentation with the final 15 minutes being reserved for Q&A. Since we have a split audience, we'll take questions from the audience here live in Chicago first, and then move to questions from the webinar audience. We will be passing the microphone around to the audience, even though it's a relatively small room here and we can probably all hear each other. That will allow the webinar audience to hear the questions as well. So thank you in advance for bearing with us on that. For those of you online, you'll be muted, so please submit your questions via the chat function. If we're not able to get to all of the web audience questions, we will log those that aren't covered and ensure that responses are provided in follow-up to the presentation today. Lastly, the slides for the presentation will also be posted on our website following the presentation. With that, I'm very pleased to introduce our presenter today, Dr. Dale Usner. Dale is president of STC and has been with our organization for the past 6 years. Prior to joining STC, Dale's experience included leading stats and data management teams at sponsor companies with AVI Bio, Zymo Genetics, and Bausch & Lomb, as well as at service providers with PPD and New England Research Institute. Dale has an extensive array of therapeutic experience and also a breadth of experience interacting with regulatory agencies for pre-IND, IDE meetings, advisory committee meetings, etc. Dale holds a PhD in statistics from Oregon State University. I've personally been fortunate enough to work with Dale for the past 9 years now, first as a client and now as a colleague. One of the many great things about working with him, and something that I'm sure you'll notice in today's presentation, is his unique ability to inspire those around him to understand the big picture and what it is that we do. And that's something that Dale instills in our entire team at STC, and kind of something that's become sort of a mantra for us internally. We actually have signs posted on the walls around our office space that simply ask ourselves the question, how do we help people? And I think that's huge for all of us in the industry, to be able to take a step back from our respective day-to-day roles and remember what we're all working towards, which is ultimately to help people by developing products to cure diseases and improve quality of life. So without further ado, I'll hand it over to our presenter, someone we're proud to call our president at STC, Dr. Dale Usner.
Thank you, Jim. So I don't stand still well, so I'm going to be walking back and forth. If I happen to stand in front of you and you cannot see, just shush me a little bit. Be good. All right. So the brief agenda for my talk today is, first I'm going to go over missing data and common analysis methods of missing data. I'll then go through a motivating example. I'll illustrate the impact of different methods in pictures. You will not see any equations today. It's pretty much graphical, very little text as well. And then I will also go over the statistical properties of the different methods that you will see, and there will be some time at the end for sure for Q&A. So missing data has been well written about over the last half dozen and more years. I have a number of references that I give here, and really what everybody's after is the understanding and the prevention, impact, and treatment of missing data in randomized clinical trials. What you'll get from agency all the time is just try not to have missing data. That's first and foremost. What can you do in your protocol to prevent the missing data from happening? And so recently I'd seen a presentation where clinical trials are getting increasingly complex year over year with respect to the number of visits subjects have to go to, the number of measures that they have to go through at a visit. All of those things are going to lead towards missing data. You're going to get subject burnout. What you see here is a list of a number of references that discuss missing data. The bottom one is was the motivation for me to give this presentation. I received regulatory feedback on a protocol that asked me to complete an analysis of missing data that I had not seen before called trimmed means. And so in looking at trimmed means and looking at the properties of trimmed means, what I wanted to do was understand the properties of that technique compared to other standard techniques, so that I understood what I was getting into in utilizing the trimmed mean methodology. Has anybody else here heard about the trimmed mean methodology? Great, I hope you're not the author. Awesome. And I've been a little sick, so my voice is probably going to fluctuate. Forgive me. So what I'm going to go over now is missing data methods, fairly common methods in both continuous as well as then dichotomous data. So first I'm looking at the continuous data, and some methodologies that you see to use observed data only. Just disregard the fact that people are missing. What is the impact of that? What assumptions are you making? What is that analysis going to look like at the end versus what happens if you actually had the data? Last observation carried forward. Okay, you've got information from that subject from the prior visit. Let's use that to impute the data for the primary visit in question. Baseline observation carried forward, which can be considered to be pretty conservative, especially if there's an inclusion or exclusion criteria around, let's say, the primary analysis, and you have to have a certain measure or higher or a certain measure or lower to be included. Baseline observation carried forward can be very conservative. I've seen that in many different types of trials. Pain trials really come into play for baseline observation carried forward. And then more recent methodology with respect to the computing processes, multiple imputation methods. There are many different models available for you to build multiple imputation methods. I'm actually not going to go into them today because they are so varied. I just could not be able to walk through them all. But within the methodology, what you want to do is set up models depending on if you're comfortable assuming that your data is missing completely at random, which means that you're going to, if you're missing, it's completely independent of any other measure in the study. The missing at random means missing data is independent of unobserved data. If you have observed data and you condition on the observed data for that patient, you can predict what the missing data actually would have been. And then the missing not at random means you cannot predict well what that missing data would have been. There are some unobserved information that would basically help you predict, but you haven't observed it. And then the method of trimmed means. What trimmed means does, and I'll show this graphically, essentially all these other techniques, they're after trying to estimate the value of that missing data point. All right, if a subject's pains, let's say a subject is in a pain study, you're on a VAS scale of 0 to 10, they're missing a certain visit. All these other methods want to try to estimate what the pain score would have been for that subject at that visit that they were missing. Is it an 8.5? Is it a 5? They want to estimate it, and then from there analyze the data. The trimmed mean method actually does not try to estimate what the data would have been. The trimmed mean method basically just says, all right, there's a reason this person is missing the value. It's either, you can say, lack of efficacy. There might be a safety issue, something wrong with the product, or the product has safety issues, and so it's not tolerable, so they drop out. The trim mean method doesn't care about that, doesn't try to estimate what the missing value is. What it does do is it essentially says those people who dropped out are the worst with respect to efficacy. And then from there, it moves forward and throws away all the worst data. So it'll, in the paper basically points to throwing away half of the data. That's what, based off of the examples and based off the models in the paper, they say that the best thing to do is throw away half of the data, the worst half of the data, and only analyze the best half of the data. So for every treatment group, let's say in this situation where you have, let's just assume you have 300 patients in a treatment group. What you're going to do is at a given visit, the primary visit, whoever is missing data, you're going to consider them the worst. And with those patients and the rest of the data, you're going to only keep the best 150 patients from each treatment group. So anybody who's missing data is thrown out. They're considered the worst. They are not kept. And I'll show that. It'll be easier to show that graphically. Dichotomous data, some methods for handling missing data, observed data only again, last observation carried forward again. The next one is missing imputed as failures. If you're missing, we're going to consider you a failure. You're in the denominator, but you're not in the numerator. That's fairly similar in most cases to baseline observation carried forward, assuming there's enrollment criteria to be in the study. And then again, multiple imputation methods. What the rest of this presentation is going to do is illustrate what the impact of the chosen technique is on essentially the operating characteristics of the model. What is your overall type 1 error or alpha, and 1 minus type 2 error or power. And I'm going to show that for different scenarios. I'm going to show it for non-inferiority tests and for superiority tests. And I'm going to show it for 3 different scenarios of missing data. One where I'm assuming that missing data is correlated with higher efficacy, meaning, all right, the drug is working, and in those subjects where the drug is working, it's like, let's consider it a sensitivity. There's something with them that they're sensitive to the drug, so they're more efficacious, but that also leads to more tolerability issues. I then have a scenario where it's correlated with lack of efficacy. Here again, I'm assuming that the product has some tolerability issues, and so what you'll see is I'm assuming in one scenario that the control group, while might also have lack of efficacy, does not have as much missing data because the test group, while it has lack of efficacy, also has some tolerability concerns, and so folks are dropping out for that. And then also missing completely at random. So the motivating example throughout is a weight loss study. This is completely fake. I'm assuming 2 different dosing regimens of the same concentration product, once a day and twice a day. I'm going to assume the primary endpoint is at 3 months and that there's also a 6 weeks visit. And the reason I assume the 6-week visit is in order to have that last observation carried forward. I am assuming that there's, you don't have to remember this, but I had to do this in order to build the simulations. I'm assuming that there's correlation between the 6-week and the 3-month of 60%. So that, you know, you would be losing weight slowly if you would. I'm assuming that there's correlation between the baseline and both the 6-week and 3-month of 30%. Again, I had to make some assumption there in order to create the data for the baseline observation carried forward. I'm going to assume that the discontinuation rate is 24% in BID. There's tolerability issues, as I discussed, 12% in QD, and 3% in the control. Further going to assume that BID is better than the control in weight loss by about 2.2 pounds, and that the QD and control are similar. This essentially leads to a change from baseline of approximately 11 pounds in the BID arm and 8.9 pounds in the QD arm and control, respectively. Standard deviation of 6 pounds, non-inferiority limit of 3.3 pounds. I am not suggesting that the FDA or any other regulatory body is okay with a non-inferiority limit for a weight loss study of 3.3 pounds. It's a fabricated example. All right, so from here on out, there are a lot of histograms. So I'm going to walk through the histograms, but also please feel free to stop me early if you have questions, because if you have questions now and don't ask, they're just going to propagate. So I would rather ask questions up front so everybody's clear, so as I walk through, it'll be a lot easier to understand. So the first example I'm going to go through is once a day versus control. Again, remember, once a day has a higher discontinuation rate of about 12%. Control was, I think, 3%. And I'm assuming that the efficacy is identical, and I'm looking at a non-inferiority case. In this scenario, the missing data is correlated with efficacy. Okay. One of the things I want everybody to understand is that efficacy is on the left. The greater efficacy subjects are on the left-hand side of the figure. The less efficacy is the right-hand side. Okay, so the x-axis here is weight. So the further you are to the left, the less you weigh. The further you are to the right, the more you weigh. All right. What I'm showing here, so in my simulations, what I did was I created full sets of data. And so what I'm showing in these two figures, the upper figure is QD. It's the once a day for the test. The lower figure is the control. The height of each of these, for each category of weight, are the total number of subjects at that weight category. And I just categorized for the histogram, understanding there's continuous data that underlines this. The orange bars are those subjects who completed through 3 months. And the green bars are the data of the subjects who discontinued. So as you can see, and as I said, I am correlating discontinuations with efficacy. And so what you're seeing here is that the green bars are more on the left-hand side where there's more efficacy. You can also see that there's more green bar on the upper figure, the QD arm. And again, that's because I'm assuming a 12% dropout or missing in the QD versus the control, which is at about 3%. So everybody understanding that all of that is very key from what goes on. The other thing you can see is that this is a single simulation. It's a single random set of data. What you'll also see is that essentially those histograms overlay each other very well. There's really no difference between those two populations. The next figure shows observed data only. So again, this is one of the techniques where you just basically analyze the data that you have on hand. And what you can see here is based off of the fact that the missing data is correlated with efficacy. Again, the upper figure is the QD arm, the lower figure is still the control arm, and you can see that in this case, although it's tough to tell, you can see that there's a bit more efficacy here on the control arm than the QD arm, and that's really because we dropped those green discontinues that are more on the efficacious side and a higher proportion of them in the QD arm. All right, here's last observation carried forward. So all those patients, all the patients in green that dropped out, I have to replace their data. They're missing. This is their actual data that I'm showing you, but I also had to generate the fact that they're missing, and then I generated their last observation carried forward, which is here. So I am filling in the data set, and this is exactly what happens when missing data is imputed. You take the data that's not there and you impute it with some information. Here I'm imputing it with last observation carried forward. What you can see in this is it really doesn't shift either of the figures too much. It looks pretty good. Here's baseline observation carried forward, which makes sense here. There was an enrollment criteria that you had to weigh so much in order to get into the study. So any imputation of the baseline observation carried forward, it's going to be on the heavier side of the figures. You can also see again here the big impact of this is that there's more discontinuations in the QD arm than in the control arm, and that really impacts these two figures with respect to, all right, now you can really see in this scenario, it looks like the control is better than the QD arm. This is one figure on the trim means, and the next figure will give you a better example of the trim means. So if you remember what I said was on the trim means, you really don't try to predict or estimate what the actual missing data was. You basically say it's just the worst data, and then you lop it off. So here I'm giving a bar on the end just to show you the number of patients who discontinued. It's not really, and I'm putting it far to the right because it's considered the worst data. So this is the first step in computing trim means. The next step is to lop off the worst 50% of the data. So this is the information that is used in calculating the trimmed means, the differences in the treatment groups in the trimmed means. So a couple of things here. So I'm going to go back. Here's the original full data. We have 150 patients in each arm. What you can see here again is there's really not too much of a difference, but what I want you to see is what is the impact of the differential assumptions on how you impute the missing data. Observed data only, not too much of an impact, but there's a little bit more or a little less efficacy in the QD arm than in the control arm, strictly because of the fact there was more discontinuations in the test arm. Last observation carried forward. I'm imputing the missing data with the data that has come before. The distributions look pretty similar to one another. Baseline observation carried forward. What you see there is it's actually fairly conservative. It really shifts that group that has a higher discontinuation rate. That histogram shifts to the right. And then the trim means you're only using half of the sample size. I say that. The authors will argue, and I agree with them, that you are using all the data because you're ordering it first and then lopping off the lower half. But essentially, when you're calculating the difference between the two treatment groups, you're doing it at only half of the data. You don't have to trim off half. That's what the authors have in their paper. There's a reason for it. Their example motivates a requirement of 50% being the best case, but it's really their example that motivates that. And in this scenario as well, you can see that the control group is slightly shifted to the left, meaning it's showing that it has possibly a bit more efficacy. Now the BID, I'm going to go through this faster now. I just wanted to go through that fairly slowly. Any major questions at this point? Am I going too slow? Okay. BID has a larger dropout rate, right? We were just looking at QD at 12%. We were looking and control at 3%. BID I assume to have 24% versus control. So you're going to basically see a very similar set of figures, but with more green floating around on that top figure. All right, so here again, dropout is correlated with efficacy. And so what you're seeing is definitely more of the green bars on the left-hand side of the BID arm. The bottom figure, the control, is identical to what I had shown you before for the control. So it's the exact same control. If you use observed data only, what you can see here, all right, in this case, because you lost so much of the BID arm and the observed data only, you're really seeing a difference between those two figures, and the control arm is showing that it's more efficacious than the BID test arm. Last observation carried forward. The BID arm, if you remember on the QD arm, when I did last observation carried forward, it was nicely distributed over the possible weights. Here there's a little bit more of a distribution to the right, and that's because when I created the data, I assumed that somebody could drop out at 6 weeks or 3 months, and this is the 3-month data. So if somebody dropped out at 6 weeks, their last observation carried forward would be baseline. So that's why it's shifted a little bit more to the right here than what we saw on the QD, because in the QD, I assumed a very small proportion dropped out at 6 weeks. Most of the dropouts there occurred at the 3-month. But here again, in looking at the LOCF, you're showing fairly well that the distribution of the BID arm is shifted right to the less efficacious than the control arm. Baseline observation carried forward, it's shifted even more so because now we're going back to baseline for everybody. So I'm going to go backwards to the LOCF and then forward again to the BOCF so that you can really see that difference. Here's the trimmed means, and what this is really showing you is that there are a lot of people who discontinued from the BID arm. It's more efficacious, but in those people where it's more efficacious, the tolerability issues are higher, so they're dropping out. And so when you take a look at the trimmed means and the distribution of the data of just the best 50%, so again, on the trimmed means, anybody who's missing, I'm not trying to estimate what their value is. I'm just saying they're the worst, whatever that is, and then I'm getting rid of the worst 50 and keeping only the better 50. And here again, you can see quite well that the control arm's distribution is shifted to the left or to the more efficacious side of the figure than the BID arm. All right, so I wanted to show that. Again, that was data correlated with efficacy and the impact of that. I'm going to now show data, again, fairly quickly, when it's correlated with a lack of efficacy. Very similar figures. What you'll see here more frequently, though, is that we're showing that those who discontinued in the full data, you're going to see that the green bars essentially are to the right side, lack of efficacy. I'm still assuming there's tolerability concerns, and that is why I'm showing that the dropout for the control is still less than the dropout for the QD or BID test arms. I'm assuming the same dropout rates or missing data rates, 24% for BID, 12% for QD, and 3% for control. All right, so if I take a look at ODO, observed data only, in this case, what happens? So here's the full data. Here's the observed data only. Somebody tell me what their thoughts are on those. What's the impact here of going from the full data and analyzing the observed data only? Right, full data looks like the figures are fairly similar. Distribution of the data is pretty similar. Observed data only, because folks are dropping out due to lack of efficacy and there's a higher dropout rate in the test arm for tolerability, you're actually awarding the test arm efficacy, essentially, for lack of tolerability, because you're throwing away their data, which makes the remaining data look good and more efficacious than control. Does that make sense to everybody? Is everybody? Okay, awesome. LOCF, what you're seeing is it fills in pretty nicely in general. You're not seeing any serious shifts in the distribution. I'm going to quickly go back to the original data. So you can see that it is on the QD arm more to the right and the heavier weights. Then when in LOCF, you are seeing that some of those patients who dropped out actually were having efficacy on the prior visit. So the distribution is evening itself out. Baseline observation carried forward. What you can see here is similar to the prior example. You're showing a bit of a shift in the QD arm towards less efficacy strictly because it has a higher proportion of dropouts. I skipped, so now my trimmed mean figures, I only have one. I skipped the first one where that big green bar is on the end. I'm assuming you recognize that by now. And I'm just going straight to the trimmed data. So here on the trimmed data, what can you see? You don't see much of a shift. You really don't. You see that those figures are still fairly similar with respect to the distribution. And again, the reason for that is I'm assuming that the discontinuation is due to lack of efficacy. The trim data throws out the 50% worse subjects for efficacy, so you're throwing out those patients anyway. And the QD arm, I'm assuming, has a higher dropout rate or a higher missing rate because of tolerability. And so once again, I'm basically adding efficacy value, if you would, to the QD arms via their, I don't want to say this, because of their lack of tolerability. Sorry, because the product is not tolerable in certain patients, those patients get completely removed from this analysis and actually benefits the QD arm. I'm going to show the same thing again for the BID arm, and then we'll go into some model properties. So very similar figures that we've been showing. A lot more green here again because it's a BID arm on the upper figure where I've got 24% discontinuation. So in this case with the full data, what we're seeing are the distributions are fairly similar with the full data. And if anything, if you remember my example, I'm actually assuming and creating the data for the BID arm as if it was slightly better by a couple of pounds with respect to the weight loss efficacy than the control. And so if I remove and just use observed data only, so I'm completely, any subject who discontinued or has missing data, I don't even try to estimate, what does that do to the figure? So here's the original data, where the figures overlap each other fairly well. And then once I get rid of the, basically looking at just observed data only, what you can see is that for the BID arm, again, which is up top, the histogram is shifted to the left, which is showing there's even essentially more efficacy in the BID arm. And that's because, again, you're getting rid of the subjects where the product didn't work and the product is less tolerable, so they quit. You're throwing that data away, and you're really only left with the subjects that were able to tolerate it and where it worked. If I fill in with last observation carried forward, here, actually, in this scenario, again, once again, it's fairly good with respect to the data that gets imputed. Here's the original. You are shifting to the right a bit from the original in the BID arm, and again, the rationale for that is a lot of those subjects are going back to baseline because I'm assuming such a large proportion of dropouts. And then if I go just to straight baseline observation carried forward, it shifts even further to the right. So in both of those cases, you're really showing that the BID arm is worse than the control arm. And if I go to the trimmed means, again, you really aren't seeing a difference in those figures. And the reason is I'm trimming off the worst 50%. And those people who tend to drop out also had tended to be the worst efficacy. So I'm getting rid of all that data anyway. So the data that's left over is very similar between the test arm and the control arm. All right. So now I'm going to talk about model properties. And essentially, each of those figures that I just showed you was a single simulation in each of these models. I ran 5,000 simulations and did a 5,000 bootstraps on each one of those simulations. It's all stats. But I basically had to create the data and then run tests on each one of them in order to give model properties. And so my model properties, what I'm going to do is walk through alpha, or type 1 error, and beta, or 1 minus beta, and power. Does anybody have questions on what those two things are, alpha and power? So alpha is the probability that you essentially reject the null hypothesis in favor of the alternative when there is no efficacy, or when the alternative is not true. And power is the probability that you're going to reject the null in favor of the alternative when, in fact, the alternative is true. Okay, so power is the probability that you successfully show that the drug works when the drug does work. Alpha is the probability that you show the drug works when, in reality, it really doesn't work. Agencies are a lot more critical of alpha. They don't want to approve drugs that aren't efficacious. Power is really a property that is more of a sponsor error rate and a sponsor responsibility, if you would. They're the ones, sponsors want to have a high probability of showing their drug works if indeed it's truly is an efficacious drug. So the way I've ordered this is to first look at alpha and type 1 error, because that's the regulatory side of things, and then also look at power to say, all right, in the scenarios where we are able to maintain the type 1 error or alpha for regulatory, what are those best methods with respect to maintaining power as well? All right, so first one I'm looking at is, again, non-inferiority. This is, for alpha, I'm only going to present the QD versus control. What I'm looking at here in this figure is essentially on the y-axis, it's the type 1 error. On the x-axis are the different types of imputations. The different colored bars are the different types of missing data. The green bar is the missing at random data, which I didn't actually present previously just because of time. The orange bars are when missing is correlated with efficacy, and the blue bar is when missing is correlated with a lack of efficacy. My y-axis goes from 0 up to 0.1. This is a one-sided alpha, so the target is 0.025. And sure enough, with the full data, when I ran the test on the full data, I'm hitting 0.25 across the board. Then again, I basically simulated those people who had missing data or discontinued and ran the other models. So on observed data only, what you can see is if you're missing at random, it makes sense. You're missing at random. If I'm going to use observed data only, I'm going to have an alpha very similar to the overall alpha. And sure enough, it hits right at 0.025 because we're missing at random. If I'm missing, and that missingness is correlated with efficacy, so the more efficacious I am, the higher the probability I'm going to have missing data. And I'm assuming that the proportion of patients who are missing data is higher in QD, 12%, than control at 3%, again, because of product tolerability. Then in that case, I am actually going to have a much smaller type 1 error. I have a lower probability of saying that the test product is efficacious, even when it's not efficacious. So it's lower than 0.025. Why does that matter? The FDA is not going to argue about that at all. As a sponsor, I would be concerned about that because if my alpha is that low, that means my power is probably also going to be lower. So I would want to attempt to use a model that gets me at about the 0.025 for alpha because that's going to increase the power I get per sample size. And then one more, is the blue is missing is correlated with lack of efficacy. So in this instance, if I'm, for observed data only, I'm only using the data that remains. So if I'm basically throwing away data that's less efficacious, then what you see is that my type 1 error is going to go up. So essentially here again, I am benefiting, my product is benefiting because it has tolerability issues that are associated with lack of efficacy. So if it's efficacious, I'm seeing I'm dropping weight and I've got these issues, I'm going to stay on it. You know, it's not enough to overcome, the tolerability issues aren't enough to overcome the fact that I'm losing whatever weight I'm losing. But if I'm not losing weight and I've got these tolerability issues, I'm getting off. And that's exactly what you're seeing here. So in that scenario, for observed data only, your one-sided alpha is actually very high. It's about 4 times the stated alpha of 0.025. Last observation carried forward. If you remember through the examples, that as a general, you know, looked pretty good on the histograms as a general rule. Sure enough, you know, much better, about the same alpha when missing at random. Much better alpha when missing is correlated with efficacy. Better, but still high alpha when missing is correlated with lack of efficacy. So you're around 0.035, so about 50% higher than it should be. Baseline observation carried forward, very conservative. So your alpha is really low there all the time, and that's basically you're penalizing your product by carrying baseline forward. Again, this very much assumes that there is a inclusion criteria around the primary endpoint, and that you have to have a certain level or higher, if you would, to be enrolled. So in this case, your weight has to be a certain amount or higher to be enrolled. And then the trim mean, what's neat about the trim mean here is random missing, actually, the alpha level is quite low. And essentially, that's because you're pulling data from all over the distribution and throwing it away. The missing correlated with efficacy, what you end up doing is you're taking those, you know, if you're dropping out and you had good efficacy, you're taking those patients and throwing them away. And so in both of those cases, you're not going to have the ability to show a difference. However, it maintains right on alpha in missing correlated with lack of efficacy. The rationale there is you're throwing away, in the trim means, you're throwing away the bottom 50%. You're throwing away those patients that aren't efficacious anyway. So it doesn't matter if they're the ones that are missing the data because you're throwing them away. And this is precisely what is discussed in that manuscript. That's the motivating example. And so that's why the properties in that manuscript look so good, is because that's the example that they're focused on. Now I'm going to go into power. And And so the first thing we're going to look at is power for non-inferiority. I say efficacy just to shorten the title. This again is missing data correlated with efficacy. And what you want to see here is that power is pretty good across the board, especially for BID, and that's because I had BID being so much better to begin with. So the QD arm is what I'm going to focus on here just because it moves around a little bit more. So my overall power was very high. This is the full data, so my overall power was at like 99% anyway. If I look at just observed data only, the power on the QD drops to about 96, 97%. So it dropped about 3 or 4% power, not too bad at all. Last observation carried forward actually maintained power fairly well. Baseline observation carried forward, if you remember, that's basically shifting in your distributions, and the power to show a difference or to conclude non-inferiority in this case drops fairly significantly. We are still maintaining an 80% power, between 80 and 85%, as you're seeing on the y-axis, but it does drop down from essentially what it would have been if you had all the data. So it drops down about 12% in the power. In the trim mean, what you see is that, and you'll see this throughout the figures, the trim mean usually falls somewhere in between the baseline observation carried forward and either observed data only or LOCF. So it's essentially more conservative than LOCF, more conservative usually than observed data only, a lot less conservative than baseline observation carried forward. And part of the thing that's sort of neat about that, I'm going to step back, is one thing that people think about when they look at the trim means is you're throwing away half the data. So I'm reducing my sample size by a factor of 2, and I'm still getting that power. What's going on? And the reason is you're actually not throwing away that data. You're not using it in the analysis, but you are using it in the determination of what data is kept in the analysis, because you're specifically ordering the data and then only looking at the top 50%. So in doing that, what happens essentially is you're really no longer looking at the normal distribution with a standard deviation of sigma, whatever sigma is. You're looking at a different distribution with a lot less variability because of the fact that you're only looking at the upper tail, if you would, of that distribution. And so that's why you're maintaining power, even though the sample size is essentially halving. Power for non-inferiority for last observation carried forward. Again, on the BID arm, because how I had it powered, it really maintains power throughout. The QD arm shows very similar, where it basically drops for both the baseline observation carried forward as well as the trimmed means. On the random side, again, a very similar outcome. I'm going to jump to the superiority data. And so on the superiority side, this is alpha. Looks like a very similar figure here, and as it should be. In testing non-inferiority, testing superiority is actually very similar. It just, at one, you're looking at sort of a boundary of 0 for superiority, and on the non-inferiority, you're looking at some boundary that is the non-inferiority limit, whatever that boundary is. But it's really similar. You're just shifting the boundary that you're interested in. And so in this case, we see very similar outcomes for the observed data only, where the random missing has the stated alpha of 0.025. When missing is correlated with a lack of efficacy, again, you're basically rewarding your product because it has tolerability issues, because you're throwing away all that data and only keeping the better data for your product. And that's why your alpha jumps quite significantly. What you're seeing then on the LOCF is also very similar. As we saw before, baseline observation carried forward, you're essentially never rejecting the null, or very rarely rejecting the null. And on the trim mean side, again, this is the motivating example in the manuscript, is missing correlated with lack of efficacy, and sure enough in that, your alpha level is maintained at right around 0.025. And the reason I wanted to come here is this, in the superiority simulations that I did, the QD was powered at right around 81%. So the full data power is showing about 81%. The BID arm was powered based off of the sample size for QD, its power was at like 98%. But what you can see here is when missing data is correlated with efficacy, so the better you are, the worse the tolerability is. Sorry, the better efficacy, the worse the tolerability. So you're actually dropping out those patients who did well. Your power drops significantly on the observed data only. When you're looking at LOCF, here again, I'm correlating it with efficacy, if you remember correctly, at a much higher discontinuation rate in the BID arm of 24% than the QD arm. And that's why the last observation carried forward. The power is actually also dropping fairly significantly because I'm taking that information that should have been very efficacious and I'm filling it in with the last visit, which wasn't as efficacious. And so I'm dropping power fairly significantly there, as well as then on the baseline observation and trimmed means are both the worst and quite low. Missing data correlated with lack of efficacy. In this scenario, what you're seeing on the observed data only, and I had shown that previously, your power actually is better than if you had all the data. So if you know your data is missing due to lack of efficacy, it actually is better for you to do observed data only than if you were to have all of your data. And that you can basically, if you compare the two green bars, you can see that the green bar is higher under observed data only, as well as the orange bar is also higher under observed data only. Again, that's due to the fact that I have a higher discontinuation rate assumption or a higher missing rate assumption in the test arms. The last observation carried forward, power is reduced. Baseline observation carried forward, again, the worst case. Here the trim mean, again, the motivating example in the paper, actually works fairly well. Your power is let's see, the power for the QD does drop down to about 65%, and the power for the BID is at about 81 or 82%. So it maintains quite a bit of power. It's not overpowered, as in the observed data only. It's much better power in the BID than the LOCF, and then also then in the baseline observation carry forward. And on the QD arm, it's in between those two. All right, and then when the data is missing at random, essentially. So in this case, when data is missing at random, what one would expect is the observed data only arm to be almost identical in power because you're missing at random. It's not going to take away from it's going to equally take away from efficacy and lack of efficacy. So your overall distribution will look the same. Your sample size reduces a little bit because of that, and so you're going to get a little bit less power, but not much. And you can see that in these comparing those two figures. And then if we move forward to the right on LOCF, BOCF, and trim means, again, what you're seeing there is a reduction in power, primarily due to the fact that I'm assuming a higher discontinuation or missing rate in the active arms than in the control arms. And that's why you're seeing a reduction and more of a reduction in power for the BID than the QD. Okay, so what does all this mean? I'm not going to go to here yet, but I'll be there soon. What does all this mean? What's the takeaway? Couple. I'll make them up. One is there's really no one best method. It really depends on why your data is missing. If there's a different, especially in scenarios where your proportion of missing or proportion of discontinuation is uneven between the treatment groups. That's the scenario where there's most concern. And then from there, the three examples or scenarios that I gave, which is, missing due to efficacy. So there are tolerability issues, and those are worse when the drug is more efficacious. Missing or discontinuation for lack of efficacy, meaning there's tolerability issues, and I'm okay with it as long as I'm seeing efficacy, but if I'm not seeing efficacy, then I'm getting out. And the other is just purely random. There's nothing really with respect to the product. I'm just random missing. So in each of those cases, and each of these methods, it just depends on what method you apply and the reason for missing on what the model properties are with respect to alpha and power. The agency is going to be concerned mostly, appropriately so, with the alpha and make sure that that's not inflated. So the key real takeaway here is this is precisely why the agency generally says, I want sensitivity analyses. Can you show me, you know, if you go and say, I'm going to impute it this way, they'll come back and say, can you also do it this way and that way, and make sure the results are the same. And if the results are different, that's when you investigate and try to figure out why. So that's really the methodology that's currently out there right now with respect to understanding of, are you just picking an imputation technique that benefits you? You saw that a couple of times on the observed data only, where that was a major benefit in situations where you're dropping out due to lack of efficacy. And I'll say one more thing there, as I had said, and let me go back here. So this is really the slide where I can, you know, if you're missing due to lack of efficacy and your alpha is that high, beware of placebo or vehicle-controlled trials where the placebo and vehicle are dropping out at a higher rate, because it is most likely due to a lack of efficacy. And so if you remove them and you're not imputing their missing data, you actually have less of a chance in that situation. So in this case, I'm assuming that the active group has a higher discontinuation, but if it's actually that the placebo group has a higher discontinuation rate and it's due to lack of efficacy, if you do not do anything and just use observed data only, you're actually hindering your chance to show an efficacious result in that scenario. So just be careful of that.
Thanks, Dale. So, like we said at the start, we're going to take questions from the audience. I think we have a few minutes anyways.
Yeah, sorry.
So I will go ahead and pass the microphone and just raise your hand if you have any questions for Dale that you'd like to clarify, or questions related to the topic.
No? All right.
Are you aware of recent examples where FDA has accepted methods of single imputation for primary analyses?
At different divisions, they still are accepting it as long as you also have a multiple imputation method or other methods with it. Yeah. Yeah. It's getting rarer, though.
You talked about the trimmed means analysis with certain skepticism. How does the FDA look upon that?
Yeah, so let me clarify. So I'm sorry that came across, but thank you for letting me know it came across that way. I actually was really excited about it when I saw the paper, because I was like, somebody came up with a great idea, and it utilizes some different statistics. So statistically and methodologically, it was really intriguing to me. And I do still think it's a great idea. I just think we have to work on maybe broadening how it's used, making it a little bit more flexible and accounting for the operational characteristics that I showed. So in certain situations, the motivating example being lack of efficacy, looking really good there. And so I think for me, it's more about, it's another sort of tool in the toolbox. With respect to another way to analyze missing data. I like the fact that I'm not trying to impute what that missing data was. I really do like that, because by doing that, I'm accounting for the fact that I don't care if it was still efficacious. The subject's dropping out possibly for tolerability or otherwise. They're not staying on study. So regardless of whether or not it was efficacious, that should be considered a failure, and it treats it like that. So I very much like it for that reason. I haven't seen it be requested as a primary imputation methodology. It was a request specifically as another sensitivity analysis, and I actually the study is still ongoing, so we haven't actually submitted the information yet. Sorry, I don't know if I answered your question at the end of all of that. So they're the ones who asked us to do it. And so whether or not I doubt they'll accept it as a primary. I think it's something that they've recently seen, and they're wanting to better understand the properties of it. The skepticism that you might have heard, and I do apologize because I'm actually really like it. It's just that the paper makes it sound really good, and I wanted to understand when it was really good and when it didn't have quite the properties. And so the lack of efficacy aspect is when it works quite well. Sure.
Continuing on the topic of trimmed means, everything that you presented was based on a 50% trimming.
Yeah.
Have you by chance looked at operating characteristics for other percentages of trimming?
Yes, I did, and I have all of that on my laptop, and it just depends on, honestly, it's going to depend on the reason for the missing data, and then the amount of the missing data. Both of those are really impact what the best amount to trim. Again, in the manuscript, they came to the conclusion that 50% is a good place to trim. The rationale for that is the way they simulated the missing data. They basically simulated it from the lower half of the distribution. So cutting it off at half is basically just, I'm sorry? Yeah, exactly. So it does nothing. And so that's why they came to the conclusion that 50% is best. And I'm continuing to do my work on looking at different cuts and when is it better to do different cuts. And it just depends on where the data is missing from. But that will be a big point of discussion with any regulatory body. If you come up and say, I'm trimming 33%, or that's probably okay, but like, let's say 21% or something, I'm gonna be like, all right, how'd you pick that, you know? Especially if it's not pre-specified in any analysis plan and you show up with an analysis, oh, look, 21%, it works. They're not gonna like that. Sorry.
Other questions? As stated at the beginning, any questions that came in via the webinar, will definitely follow up with responses to those. And I guess, first of all, thank you, Dale. The slides will also be posted on our website. Of course, the purpose of this presentation today was educational, but I wouldn't be doing my job if I didn't mention, if anybody is interested in speaking with SDC regarding our services, you can definitely do so. Those of you in person, feel free to either stop by our booth, talk to us after the presentation, or of course, follow up via our website or otherwise. So thank you everyone for joining.
And feel free to follow up with questions if you didn't ask them here. Feel free to send them.
Thank you.